So, thank you all very much indeed. Thank you, organisers. Thank you for all for coming. What an honour and a privilege to start off a meeting like this. This is a lovely place. This is what I saw yesterday when I arrived here. I want to talk about pathfinding. It's a sort of ideas rather than microbiology-based talk, and I hope I'm allowed to show some cancer cells at the beginning. But the ideas are absolutely fundamental to what lots of microbes do, and I hope you enjoy it. So I want to start with this famous old movie taken by, I think, David Rogers in the 1950s. Astounding that people could get images of this quality in the 1950s. Really, it was so difficult compared to now. But what this is showing is a human neutrophil chasing a bacterium and chasing it and changing shape until it gets it. And everyone loves this movie because it's so anthropomorphic. You know, you can smell the neutrophil wanting the bacterium, and you can smell the bacterium wanting to run away and squeaking as it does so. Actually, this has the whole encapsulation of cell biology and the sort of thing I'm interested in. I've spent most of my working life trying to understand eukaryotic chemotaxis. And what you see with this is how important space and time and organization and the control of shape is, and the sort of thing that tends to get lost in this world of pathways and oversimplification, because you have to consider space and you have to consider time, and if you don't, you won't understand this. Now, the story I'm going to tell you starts with melanoma. It starts with melanoma and a clinical fellow. I have a lovely pointer and it stopped working. Never mind. I'll use my arm. It starts with melanoma. The interesting thing about melanoma, the frightening thing about melanoma, is it always spreads. And what you see when you look at a melanoma like this one is you see that it is deliberately spreading with inflammation. Because if that cell that started this tumor in the middle just started growing and growing outwards, and the cells just started moving randomly outwards, then it would be shaped like a bell curve. It would be Gaussian, with the middle where it started having the most density and a decreasing density of cells going outwards. But what you can see is it's a ring. There's a higher density of cells outside and less inside. So what you can tell from this, and what Andy there showed, is that these cells have information. They know where the inside is, and they know where the outside is, and they are going from the inside of the tumor to the outside, to the rest of the body, and that's why they spread, and that's why they're frightening. But the really interesting thing about this is, in order for them to know where is inside and where is outside, there has to be information. Somebody has to make a signpost, and we studied this. So the first thing is, how do the cells move from the inside to the outside? And the answer is, of course, chemotaxis. That's why I'm working on it. So if you set up a chemotaxis assay where you put cells here and serum here and start it running, then you see something that is actually really quite frightening. When we started getting these results, I actually went to the doctor, got all the dark spots on my body checked out because they scared me. If the cells do this, they're going to kill you in a very small number of weeks. So you see they migrate in this very, very directed way towards the serum gradient. What is it in serum that they are moving towards? Actually, it turns out to be a molecule that many of you who work with immunology probably know. It turns out that those cells are uniquely migrating towards LPA, lysophosphatidylate acid. So if you take some cells doing the same migration and you compare them with cells where the LPA receptor is inhibited, you find out that blocking the LPA receptor doesn't hurt the cells, doesn't stop them growing, doesn't stop them moving, but their direction is completely lost. So the information in tumors turns out to be something to do with LPA. And to our amazement, we found out when we looked at mice that were prone to melanoma, and had therefore been killed to prevent them suffering, if you went and cut out those tumors and you cut a little section of tissue from the middle of the tumor and the edge of the tumor and the outside of the tumor, you actually found out there was a gradient of LPA in every tumor we looked at. So the instructions for the cells to get from the tumor to outside, the rest of you, turn out to be a gradient of molecules such as LPA. But this begs the real most interesting question, which is, why is that gradient there? Why does the body choose to make a gradient of LPA or similar in order to get melanoma cells out from the tumor into the rest of you to kill you? It's mad. And it turned out that that was a very good question to ask, because it turns out that the thing that sets up the gradient is the melanoma cells themselves. I'm going to have to use the laser pointer. If you look at the cells themselves, they don't make any LPA. But if you quantitate the amount of LPA there is in rich medium and watch what happens when you put cells in it, then dense cells destroy it all in about 24 hours, and less confluent cells take much longer. So cells are very, very good at breaking down LPA. And this led us to do an experiment that no one had ever tried before, because it would be weird to, where you didn't put a gradient on the cells. So again, we've put cells here, and we've put no cells here, but we've just put homogeneous rich medium everywhere. And what happens? It's absolutely identical to what happened before. In fact, the first 6 months or so I was giving this talk, I had absent-mindedly swapped the give the cells a gradient and don't give themselves a gradient slides because they're so similar. So if you put melanoma cells in an assay like this, then the melanoma cells themselves will create a gradient of LPA, and then they will migrate up that gradient, which is extraordinary. We always think of things in sort of simple terms. Someone puts in the gradient and the cells respond to it. But in this case, they're doing it. They're making the gradient while they respond to it. Hence, this idea of self-generated gradients. The cells make a gradient while they migrate up the gradient while they make it. So we got very interested in this in a theoretical sort of a way. We started thinking about how might self-generated gradients work, and we started thinking about where might we find self-generated gradients. So what I'm going to do for the rest of this talk is jump higgledy-piggledy between models, microbes, E. coli and Dictyostelium, and cancer cells. And forgive me if I don't always explain what they are, because it's the idea rather than the particular cells that are interesting. Here are the people who've done all of this, and it is an absolute mathematician's paradise, because you can only really understand it by considering positive feedback loops and differential equations. But the results are understandable and relevant to all of you and everyone. So these are the collaborators who have done this. They're a mixture of mathematicians converted into biologists and still mathematicians like Matt and John and engineers like Michele. Okay, let's start with a model. This is a model showing what these cells are doing when they self-generate a gradient. So you see lots of red stuff, which is an attractant. You see the cells breaking down the attractant, and as they break down an attractant, they make a gradient and walk out towards it. And it works absolutely wonderfully. It's robust. It doesn't mind what parameters you put in it. You can change the density of cells. You can change the density of attractant. It pretty much always works. So it's going to be a very useful scenario to imagine in biology. But then you ask, where would we see this in real life? And I'm pleased to report to a microbiology meeting that the first place it was seen in real life was in E. coli by Julius Adler in 1966. and I'm not the youngest person in this conference, but in 1966, I wasn't walking yet. I was there, but not walking. And Julius Adler showed this extraordinary and pretty much forgotten thing, whereby if you took cells in soft agar that was just galactose rich, then they would make a ring, a so-called Adler ring. And what is happening here is exactly as I showed you in that computational model. You put the cells there, the cells are eating the galactose, and as they eat the galactose, they create a gradient, and that gradient is unbelievably local. So outside here, there is lots of galactose, and inside this ring, there is none at all. So you have a constant dynamic gradient where the galactose is diffusing that way, and the bacteria are eating it, and the bacteria are slowly migrating out as they use up all the galactose. More amazingly, if you use a much richer medium, like tryptone agar, you get 3 rings. And the outer ring there is a ring of a self-generated chemotaxis towards serine. And outside there, there's plenty of serine, and inside there, there is no serine. So that ring has done it. And then you see another ring of aspartate chemotaxis, and another ring inside that of threonine chemotaxis. So all of this was shown in 1966, and then by most of the world completely forgotten as we went to simple models of chemotaxis. Amazing paper. Here are some Dictyostelium amoebas. Now, folate is the chemoattractant they use to chase bacteria. And what you see is, again, this is a slab of agar that's got a homogeneous amount of folate in it to start with. And what you see is the Dictyostelium, thinking that they are chasing a gradient of bacteria, make this wave of cells. We've used an algorithm here to put a bubble around them so that we can see whether they're moving forwards or backwards, shown in red, or the blue ones are just jiggling. And again, you see this beautiful wave of cells that are making their own gradient of folate and then chemotaxing up it. I showed you this melanoma slide before, but we've found numerous types of cancer do exactly the same thing. Pancreatic cancer, one of the most feared and spready cancers, makes its own gradients and migrate up it. We're just working on glioma, which is another feared and spready cancer, and it's entirely clear that glioma is spreading itself through your brain by making self-generated gradients of attractive molecules. So these things are everywhere. We must understand them if we're going to understand why cells go where they go. Why would cells want to use this slightly funny feedback loop-driven thing? Well, it turns out it's much the most robust way of moving around. So if you consider some cells that are moving up an imposed gradient, or some cells that are moving up a self-generated gradient, in each case, there's a source of attractant here, and we're putting the cells there. So here you will see the gradient evolve this way as it sets itself up. Here you will see the cells set up their own gradient. So in the cells that are responding to the gradient that you've given them, they do very well at the start, and then they get less good as they get higher up the gradient. And this is obvious. This is what we should always have expected, because at the beginning, there's no attractant at all on this side, and there's some attractant on this side. There's a complete difference. But by the time you're getting up to this end, you've got fairly high chemoattractant on this side and slightly higher chemoattractant on this side. There's less information, so the cells obviously do less well. In a self-generated gradient, like you saw with these E. coli in their rings, or like you saw with the cancer cells, you see that it's very, very local. The gradient is very, very sharp, and it is self-sharpened. The math of this works out that if the gradient starts getting diffuse, the cells will sharpen it. So the amount of information is humongous. The cells can align themselves much more accurately than they ever could by a gradient that you've imposed from without. As gradients get longer, this one here is now 1 millimeter, it gets clearer and clearer and clearer. So it turns out that 1 millimeter is about the most you can ever possibly navigate on an imposed gradient. So there are normal cells modeled, and they do the first half all right, and then they get lost as the gradient gets flatter and flatter, and you find they never be able to chemotax up a 1 millimeter gradient. Whereas in the self-generated gradient, they're making it themselves. They're carrying it with them. They can go on as long as there is enough cells and as long as there is a source of attractant. They don't mind. So to prove this, we built a 1 centimeter chemotaxis assay with some dictyostelium cells. You can see it's tiled up because we didn't have a microscope that would image a centimeter all at once and show the cells. And what you see is they don't care. They start off chemotaxing at the beginning. At the end of this experiment, they were chemotaxing just as well as at the beginning because they're making their own gradient. They're carrying it with them. It really is not an issue. So if a biological gradient is long, and quite a lot of the biological gradients that you folks are interested in will be long, then it must be made by the cells organizing it themselves. It doesn't work if somebody else does it from outside. But then you start getting interesting things like decisions. So if we set up a situation where a group of cells there have 2 different places they can go, you can see that when they come to the decision point, they split in 2. And actually, they split exactly in 2. This is not really a stochastic process because the supply of attractant is 50-50 from the 2 wings. So if more cells start going that way, then that arm is going to start being more attractive. It'll have more supply. So you can make a slightly more complicated decision where this end is connected to the outside world and this end is not. And you can see that more cells start going in the arm that is connected to the outside world. And then you get an amazing one. This is mind-blowing when you first see it. If you have a slightly shorter arm here, and this one is connected to the outside world, then most of the cells just choose to go this way. They don't even need to go in here at all. So they can see ahead of themselves by depleting a chemoattractant. It's a most surprising result, a most unexpected result, and we see it more and more, and it gets more and more complicated. So if you build a maze, I'm showing lots of models here. How many people are happy with me showing models, or how many people would prefer me just to show cells? Can I have a show of hands for people who like models, and a show of hands who... Lots, very good. Actually, how many people would prefer cells first? About the same, maybe slightly less. Anyhow, never mind. Sorry about that. Here's a model. What you see is that the cells break down the gradient, the cells break down the attractant, sorry, the cells make a gradient, and they never go up the blind junctions, even when the blind junctions are bent and they can't see the end. So the fact of depleting an attractant is sufficient to get information forwards. The cells can see where they ought to go based just on depleting an attractive molecule. So for those of you who like reality, I'm delighted to report that reality is quite a good approximation to my nice models. Real cells, these are dictyostelium, do almost perfectly. I mean, of course, we knew they would because the parameters for the model were all measured from the dictyostelium. But cancer cells do the same. I dare say bacteria should do the same. Maybe we should stick some chemotactic bacteria down a maze, Judy. What do you think? Down a maze. So this is as biologically relevant as you could possibly want. You think when immune cells, when neutrophils have to extravasate from your blood cells, from your blood system, and go and find an infection, what they're doing, getting out of the capillaries and between all the cells, is a maze. And embryonic development, when the germ cells have to move from outside the embryo all the way and find the gonad, it is a maze. It is a complicated path, and they must be reinforcing it as they go. And it turns out that a lot of the genes used by, for example, Drosophila are connected with making gradients. Neural pathfinding. We know that when nerves connect to one another, they use chemotaxis. But imposed gradients where the targets secrete something are unfeasibly complicated, whereas self-generated gradients can actually really quite nicely explain neural pathfinding, et cetera, et cetera, et cetera. Wherever a cell needs to get from A to something else, self-generated gradient is a really nice way of achieving it. Some of you may have seen Physarum on the news solving mazes, and I'd just like you to know this is magnificent science, but it is totally cheating, it is totally disgraceful, because as you see, when they do these mazes, and these mazes are huge, but when they do these mazes, they start by having the Physarum everywhere in the maze. And what you can see, if you look at it, is not only are the Physarum cheating by being everywhere in the maze, but they also cheat by walking over the top of the maze and other such things. So the thing that Physarum does is marvelous. It can find the shortest way, but it solves the maze by going simultaneously to all branches and then contracts into the best route. Whereas the cells that I've shown you work out how to solve the maze using chemotaxis and the breakdown of attractants and self-generated gradients. When the mazes get more complicated, it still works, but you see a more interesting behavior. So in this maze here with more structure, at first you see the cells splitting 50-50, and then you see these cells depleting everything here, and then everyone else goes well. And there's some quite complex math which we have worked out. The paper is being submitted as we speak on what the ideal path lengths and conditions are, but needless to say, we can work out how cells do this, and real cells will do exactly what the models predict. And we can actually use analytical maths to determine which path most cells are going to take. Finally, one of these mazes is easy, and one of these mazes is hard. And we know which one is easy and which one is hard because we designed it, because we used models like this to work out the principles. But how many of you can see which of these mazes is going to be easy and which is going to be hard? Anyone? Give me a sign. No? Okay, let me... So in this maze, the decisions are consistently between lots of equal branches, while one of them is connected to the next step. In this maze, the connection is between a single long pipe and something complicated. And the trouble with this one is that the rate at which attractant can be supplied down this pipe is less than the rate at which attractant can be supplied by that big thing. So where the cells in this one nearly always make the right decision, look, there they are, they're all going to go up here. You lose some of them, but by and large, they can work it out. In this one, they're going to prefer to take this branch because the supply is greater. And lots of them will go off this branch, and most of them will go down that branch. So although these mazes look identical, we know that this one is easy and this one is hard. And although the cells find it a little bit harder when the mazes get this complicated, by and large, they do just what we'd expect. Most of them make it through there. These ones constantly go the wrong way, so no one, oh, someone did. But each time there is a decision to be made here, they tend to go wrong. Each time there's a decision to be made here, they tend to go right. And actually, if you analyze the way this works, you find that the diffusion coefficient of the attractant is terribly important. And the time the cells have to see the path is terribly important. And you have a sort of a patch here where the cells will actually make a wrong decision. Down here, the chemoattractant diffuses so fast that if the cells can deplete it, they can drain all the side branches of the maze, so they can work out the correct way to go every time. But if the diffusion of the attractant is slow, or they don't have too long to work on it, then they will go the wrong way. And so in these mazes that I showed you, in the easy maze, nearly all of the cells went right. That's shown correctly, I should say. And very few of the cells went the wrong way. But in this maze here, you actually get a mirage where cells think they should go the wrong way in the maze. Okay, I've said enough. My time is up. I hope I've kept to the right length of time. What I want to do is I want to leave you with 2 conclusions. I'm trying to remember what they are now. The first conclusion is that we really have, in this day and age, to go back to mixing experiments with cells, models, and equations, and feeding them all back and forth on one another. And by doing that, you can understand things that are much more interesting. And that leads on to my second conclusion, which is that complexity is your friend. Complexity shows you interesting biology. And there has been this insane fashion in biology this past 20 years to try and say everything is simple and try and neglect spatial things and temporal things. And as you see from these mazes, actually, the most interesting biology comes when you embrace the complexity and you love it. And the tools are better than they ever were. The modeling is wonderful, and the engineering is wonderful too. And I strongly recommend that everyone tries to put the two of them together. Thank you very much indeed.